Hey guys, lecture 2 on cell transport. We're going to start off with a little bit of a review of osmosis. Remember that if I threw a red blood cell into a bucket of water, what would it do? Well, the tonicity of the red blood cell will be higher than that of the water, which means that the water concentration would be higher in the bucket than in the cell. So where would the water go? Well, it would go into that cell, and it would make it swell and swell and swell until finally it burst or underwent plasmolysis. And that has to do with this concept called osmotic pressure, ties in tonicity here, and of course this is what we did our lab on. On the other hand, if I took a red blood cell and I threw it in a bucket of salty water or pickle juice, what would it do? Well, it would shrink. It would lose its cellular contents in a process called crenation. It would shrivel up just like a pickle. And so remember that in short, when we talk about cellular transport, we talk about passive processes. Remember, it's with the gradient always, whether we're talking about the smoke particle concentration or water concentration. It moves from areas of high concentration to areas of low concentration, requires no energy, obeys a concentration gradient. Passive transport, boom, it's easy. Now, active transport, on the other hand, uses energy and moves materials against the concentration gradient. Okay, so we have a couple animations here. They're showing the movement of materials against the gradient, sort of. But listen, when you think about active transport, you probably are going to think about the sodium-potassium pump, also known as the NAK ATPase. It's an ATPase, which means it's an enzyme that breaks down ATP. And when it breaks down ATP into ADP, it moves sodium and potassium. It does it in a weird way. It throws 3 sodiums outside of the cell and it imports 2 potassiums. And you find the NAK ATPase in tissues like muscle and nerve cells. And notice that they're creating a concentration gradient. There's a lot of potassium inside the cell, but I keep moving it in. And every time I move potassium in, I use ATP. Boom, active transport. Up the gradient using energy. Now, this is a good example of active transport, but when it comes to like really using energy, we have to talk about mass transport. So, cells move materials using vesicles. How do I get glucose into a cell? How do I get large pieces of proteins for digestion? Well, the answer is vesicalization. And I can sort of do 2 things with the vesicle. I can take them in or put them out. And we call these processes endocytosis and exocytosis. Endo means in, cytosis means cell. So you can see here in this animation the movement of materials into the cell. On the bottom animation, I see exocytosis. If you look closely, you'll see some secretory vesicles that dump their contents into this space. This is actually what we call the neuromuscular junction. Here's the nerve cell with its terminal bouton. In the interior, we see secretory vesicles that contain neurotransmitters. When an electrical impulse comes down here, it causes the vesicle to fuse. Neurotransmitters are released. They diffuse across here and tell that muscle cell to contract. All right, endocytosis means to take in. Exocytosis means to put out of the cell. Let's look in detail at endocytosis. You can see this tiger is trying to endocytose this mahout. All right, and when we talk about endocytosis, we talk about phagocytosis, which is when you take in solid substances. These 2 guys aren't too happy. They're getting eaten alive. So, when you think things are going bad, imagine being this guy or that guy. All right. Panocytosis is the endocytosis of liquids. All right. Cellular drinking, panocytosis. Last but not least is receptor-mediated endocytosis. And this is exactly what we saw in this slide here. Here's receptor-mediated endocytosis. This is how glucose gets into the cells mostly. I know I showed you the glucose uniporter, right? But that was for glucose that was moving with its gradient. What happens if I don't have enough glucose? Well, this is how it happens. Receptor-mediated endocytosis. Here's some static diagrams for you to look at. Some fluid gets pinched into this little invagination, and now I've got a vacuole or a vesicle. Same thing for solids. The cell will wrap itself around that solid, make a vesicle, and then take it in here, maybe to combine with a lysosome. On the other hand, we have exocytosis. And again, our perfect example here is at the neuromuscular junction, right? The cells of the nervous system make all kinds of neurotransmitters. You know, neurons are fascinating. They communicate both electrically and chemically. And in this case, an electrical signal comes down and it tells this cell to release some chemicals. Those are proteins made all the way up in the cell here, and they're released to send some message. There's lots of examples of exocytosis of neurotransmitters or even hormones like insulin. Remember that the pancreas has specialized cells called the acinar cells, and particularly the acinar beta cells. They're constantly monitoring blood glucose levels, and when blood glucose levels get too high, they release the hormone insulin. Now, insulin travels around, and every time it binds to a cell, it says, hey, let's get rid of some of this glucose. So if I eat a candy bar, my blood glucose levels go up, and I don't want sugar hanging around in my capillaries because it causes damage, and so I got to get rid of it. I need those cells to take it up so they can do cellular respiration. So insulin's the guy that sends the message for cells to take up glucose. Now that's it. So active transport, we use energy. Remember, every time that vesicle gets moved, remember we move that with kinesin molecules along microtubules, and that takes tons of energy. Tons and tons of energy to exocytose or endocytose materials, all right? So overwhelmingly, mass transport uses way more energy. But this idea of signals and hormones and messages is really important for our next unit, or cell communication. Now, remember, cells communicate with one another. They can communicate right with their neighbor next door. You know, one cell can manufacture some metabolite and exchange it with its neighbors, probably how mitochondria and chloroplasts got together. I can have an example like that in the nervous system where one neuron is connected to another neuron, or one neuron is connected to a muscle cell, and they're connected directly and they're communicating directly. Or I can just send a hormone like insulin out and it can target lots and lots of different cells. So I can have short distance signals and I can have long distance signals. And one of the ways we have long distance signaling is through the process, or through molecules called hormones. Hormones are chemical messages that travel. They're signals, and they have a target. And their targets are receptor proteins found in the cellular membrane. If you'll remember from early in this unit, we talked about proteins that are integral to the membrane. One of those classes of integral or intrinsic membrane proteins were receptor proteins. So hormones like insulin can target their receptors, and when they bind, they send some special signal to that cell. They say to them, hey, take up sugar, or hey, contract, or hey, get excited, or hey, watch out. It's Thanksgiving. You know, one of the things that happens during Thanksgiving is you eat a lot, and then what do you want to do? Well, what most people want to do is lay down and go to sleep. And one of the stories about that has to do with the molecule called tryptophan. Now, tryptophan gets converted in your body to 5-hydroxytryptophan, which gets converted into your body into a neurotransmitter called serotonin. Serotonin is an inhibitory neurotransmitter. So when you eat a bunch of tryptophan-rich foods, it makes you feel full. In fact, serotonin is the satiety hormone. It makes you feel full. And while it's an important central nervous system molecule, most of it's made in your gut. It tells you when to stop eating. It also is associated with depression. So if you don't have enough serotonin, you feel anxious. Kind of the opposite of what you feel like after you've had a nice turkey dinner. And of course, targeting serotonin has been a big goal of the pharmaceutical industry. Humans also, since we're animals, we also respond to external signals like light. You may have noticed that you know, as we move into the fall and into winter, you kind of don't want to get out of bed in the morning, right? It's harder and harder. And that's because your body's responding to lower and lower light levels. And this results for a lot of people in something called the seasonal affective disorder, where you have this mild depression associated with the change in seasons. Affect means your mood. And hey, guess what? It's associated with serotonin. So here's an interesting question. Do we eat turkeys in the end of November just to kind of lift our mood a little bit? I don't know. Maybe, but it's something to think about. So hormones have receptor protein targets. They're the signal. Here's the target. Again, this is the neuromuscular junction. You see the terminal bouton of this neuron. It's got synaptic vesicles here, and they contain neurotransmitters. At the neuromuscular junction, the neurotransmitter is called acetylcholine. When it's released, the acetylcholine diffuses across the membrane, binds receptor proteins here, and tells that muscle cell, hey, contract, contract. And there's acetylcholine in your central nervous system. When you take stimulants like coffee or amphetamines, you also have excited thoughts and mannerisms, all right? So if you drink a bunch of coffee, you'll get the shakes. Why? Well, because you've increased the amount of acetylcholine here, and you'll be causing uncontrollable muscular contractions. So there you go. There's some receptor proteins. They have targets where they bind, and there's a binding site on receptors that's perfectly designed for the signals. And not all hormones are proteins. Insulin's a protein hormone. Adrenaline is a protein hormone. Serotonin can be considered a nervous or neuropeptide hormone. But we also have steroid hormones. Remember that testosterone and estrogen are these fatty hormones that are produced by cholesterol, and they bind receptors too, but they'll bind protein receptors. Remember, testosterone is that male hormone. It actually tends to increase the release of growth hormone, so you get bigger, but it also makes you more muscular and more aggressive and hairier and have a harder time concentrating. And so I can have these hormone signals that target receptors all over the body, and when they bind their receptor, they cause a change in shape, and then something happens inside the cell, right? So there's a response to the signal, and those responses can be categorized a couple different ways. One of the responses to a signal, let's say insulin, is to change the permeability of the membrane to something. So, for instance, when insulin binds a cell, it says, hey, let in some glucose, right? Permeability has to do with the ability for materials to move into a cell or out of a cell. And so insulin increases the permeability of glucose for the cells. And a target signal can also activate enzymes. And that doesn't fall into the scope of our course right yet, so don't worry about that. But the most common effect of a signal molecule is to activate some sort of second messenger syndrome, that is, system. So, for instance, insulin binds a receptor protein on the surface of a cell. It doesn't shout out, hey, take up glucose. It causes an effect in the interior of the cell. All kinds of different protein complexes get moved around, energy is used, and that second system, that second series of cascading events is what we mean when we say second messengers. It's the most common response to signals. Well, that's it. I hope you've enjoyed cell transport, and good luck on your test.